Excellent. Welcome back to all the speakers. Good to see you live. Dr. Kevin Harold from Yale, Dr. Rickles from University of Pennsylvania, Dr. Greenbaum from Benaroya Institute in Seattle, and I'm missing somebody. Where's Stephen Guff? We've lost Stephen. Anyway, he'll come back. I'll also welcome Dr. Mark Piekman, working with Sanofi, responsible for the immunology section, Dr. Lisa Janov from FDA, and Dr. Peter Moll joining me and Dr. Beeston here for the panel discussion. We have plenty of questions, as you saw, and Patricia and me, we will fire them at you. But before we start, I want to pick up where Stephen left. So he said clearly type 1 diabetes is an autoimmune disease where we have this lethal dialogue between the immune system and the beta cell, but where we all agree, I believe, that the primary pathological feature is the destruction of this fantastic beta cell, the only cell in our body able to secrete insulin in a glucose-dependent manner. And we're all in this pickle because we're not able to visualize this beta cell properly and to see what is happening. If it were a red joint or a painful joint, we will not be sitting here discussing how to measure beta cell loss. And so I want to kick off by asking you whether you agree with what Dr. Pluchino launched and what I believe Carla also took over, namely that we need a surrogate endpoint. We need a validated surrogate endpoint. And you, Carla, agreed with Dr. Pluchino that C-peptide could be a surrogate, a validated surrogate endpoint. So, Carla, I will come back to you with this question. Is C-peptide a validated surrogate endpoint? And then immediately, how to measure it?
So thanks, Chantal, and thanks everybody for this meeting. It's funny to have this mix of live and not. I would say two things. I'll answer the second one first. It is an extraordinarily well-validated measure. It's important to recognize it's been done now for decades. We did comparator studies. We've done it in various ages. It's been in many locations, academic centers, private practices, etc. So there's no question that MMTT-stimulated C-peptide is an extraordinarily well-validated measure. And C-peptide itself as an assay is also a well-validated assay. So I think we should discard that. That should be accepted. So the real question is C-peptide the outcome for trials? And I think it comes to the question, what is type 1 diabetes? Type 1 diabetes is destruction and dysfunction of the beta cells. This is a measure of destruction and dysfunction. Disease-modifying therapy, the whole aim is to save the beta cells. And so therefore that would be the outcome that is directly related to the therapy that we're attempting here.
Okay, but how will we measure it? And I know that for Patricia, this is one of the key important questions also. How good are the assays validated that allow us to measure C-peptide? I heard a lot of statements. I heard 90 minutes. I heard peak C-peptide. I heard area under the curve. We need the 4 hours. So again, I will come back to Kevin now, incoming chair of TrialNet. What is the best measure of this C-peptide?
Yeah, thanks for the question, Chantal. I mean, I think it's a more complicated question, even because we do one thing in the pre-diabetes period and we do another thing in the post-diabetes period. Frankly, I don't think that I see huge issues with standard mixed meal tolerance tests, to be quite honest. It measures a physiologic stimulus, a mixed meal. It's not a strange stimulus like glucose. It's pretty reproducible in the same individual over time. Whether we do 2 hours or 4 hours is a matter of an ongoing debate that's been going on for years. I think the comment that Dr. Goff mentioned that it doesn't a shorter test doesn't completely reflect the physiologic response. I think that's true, but we can on the other hand, 2 hours really captures the majority of the response. So I don't think it's that complicated. A measure. I think a good. Now, Carla has also pointed out many times, and I think she's right. And our colleagues in Europe also do more provocative tests where they will stimulate insulin secretion even further with arginine or other measures. I think those are all fine. And on the other hand, we adopt certain ways of doing this because of feasibility. And I think for most individuals and for most studies, a mixed meal tolerance test is pretty good. And I think can be used, very easily. Can I mention one thing about a point that Carla made and also that Dr. Goff made, and I think warrants maybe a bit of clarification, which is that type 1 diabetes is not just a disease of the immune system. That's true, but I think what Dr. Goff pointed out in his comments, which I think should not be misinterpreted, is this is not a disease of insulin resistance or other dysfunction of insulin in the periphery, like type 2 diabetes. As Carla emphasized, this is a disease of beta cells at the end of the day. Now, Dr. Goff's point about it also involving beta cells in a sort of a crosstalk or a combined assault on insulin production by both the activity of beta cells in stimulating the immune system, and in contrast, the activity of the immune system in killing beta cells is very true. But I don't think we ought to be misled in thinking this is anything but a destruction of insulin-producing beta cells.
Okay.
Chantal, if I could jump in 1 second, sorry, one question. I think it's important not to get bogged down in should it be AUC, should it be peak, should it be 90 minutes? These are super highly correlated variables, and it's really a pragmatic issue rather than anything else. So just to throw that in there, that any of those measures have been well validated and standardized and reproducible in individuals.
Okay, so I think a clear message from the group of scientists to FDA, whatever FDA would think the most robust of the measures. But Patricia, you have more questions in that area.
Oh, so not particular questions, but I wanted to offer the comment that there's a difference between a validated assay, that it meets certain performance criteria and standards, and what's a validated surrogate endpoint, and there's regulatory considerations for that. So it's important that people consider that, and that's the purpose of this meeting today, is to hear what people have to say, what they have to offer, and then be able to help make regulatory decisions, and we'll have conversations. Did anybody, Lisa, do you want to add in, or?
Peter Mol wanted to comment.
Yeah,
Sure.
Sorry, I had actually a question for clarification from the scientists, because in the field of GFR slopes, for instance, where I'm a little more familiar, I know there's a lot of individual variability, and that requires that we also collect sufficient amount of data, also in individuals. I didn't hear anything around this. I assume that the issue is not that relevant in this setting, but could you please speak to that?
Carla?
Yeah, so we published on that. It was actually a combined study with Europeans and US, so it was a very large study looking at the intra-individual variability at very low. In other words, the R-square reproducibility is more than 0.9, and I'm happy to send you the reference on that. So it's extraordinarily useful and reproducible measurement.
Can I go back to what Dr. Rickles
This is Lisa.
Sorry, Lisa. Yeah, go ahead.
I can complete. Yeah. Patricia asked if I had any comment on her comment, and I wanted to add that I agree with her and just expand a little bit that when we're talking about validated in the sense of a surrogate, it's different from what Carla was discussing, which I completely agree with in terms of the assay. We know a lot about the assays, although I think there is still work to be done with the assays, but we may be close on that. The issue with validated surrogate is that we need to know whether manipulating the surrogate or intervening on the surrogate produces the desired outcome. And that's what we're missing today with some of the presentations. We saw a lot of correlational data where patients in DCCT who had preserved C-peptide were experiencing better outcomes than patients who didn't. But DCCT was able to validate hemoglobin A1C as an endpoint because it directly intervened on A1C and showed that by lowering A1C, we saw clinical outcomes that were beneficial to patients. That's not what we saw here. We didn't see an intervention that actually lowered C-peptide and produced those outcomes. We saw correlational data. And so that's what we mean by it's not yet validated.
Yeah,
Can I
wait, Carla.
Dr.
I believe
Rickles.
Mike. Yeah, exactly.
Yeah.
So I believe that Mike.
Yeah.
Yeah. Okay.
Go ahead.
So yes. So
aside from the correlative data, there's direct data that shows an intervention improved C-peptide because that's all that was changed, and it had the correlates. Once that was done, the benefits to the patients. That's why Mike Rickles' data is so compelling. So Mike should speak to that. Sorry, Chantal.
No, I mean, you're a good chair. So, Mike, I believe you have the evidence that if you bring in new islets and measure C-peptide as the product of the islets, you do have dramatic effects on all kinds of clinically relevant measures. The conundrum, however, is that you show the difference with a status when they have zero C-peptide. And then you bring in some C-peptide. And our problem is that in this first year, as Carla showed, everybody still has C-peptide. But you show that bringing in functional islets and the C-peptide makes all the difference. Am I right or am I wrong?
That's right, Chantal. Can you all hear me? Yeah. Okay. So I fully agree. The advantage we have with the islet transplantation experiment is that individuals with very low or undetectable C-peptide, upon restoring what's been demonstrated correlatively in type 1 diabetes to be consistent with clinically significant C-peptide, does immediately protect individuals from developing hypoglycemia, improves their glycemic control, and is associated with the reduction of insulin requirements that, depending on the amount of restored C-peptide, may allow for normal glucose homeostasis and insulin independence. And I think what's really remarkable is that in the data presented, the restoration of levels of C-peptide by islet transplantation match almost identically that in residual C-peptide in type 1 diabetes in terms of their correlations with measures of glucose control and avoidance of hypoglycemia. So it does seem to speak to the effect that you could anticipate similar benefits of preserving that level of endogenous insulin secretion than of restoring it in individuals who've already lost all ability to make and secrete insulin.
So I would like to ask a follow-up question to that point. So the islet cell transplant is not merely giving beta cells. You're also giving alpha cells, or you are restoring some, glucagon function and counterregulatory hormones. The other is if they have a preserved C-peptide, that might indicate that the islet cells have not been totally wiped out, and you are still maintaining some benefit of within the islet balance of insulin and glucagon signaling. So I'm not sure that to say, oh, if you have C-peptide is doing something. C-peptide, like we're trying to discuss, may be a marker of something that is happening differently. So if you had an intervention somehow where you could only improve C-peptide in patients who had a prolonged duration of insulin independence and have no measurable C-peptide, but the only thing you gave them back was restored beta cell function, you may not get that same benefit because you're not giving them all that same supplementation that you would find in an islet cell. So I'm just trying to be very careful about how we are interpreting it.
Well, I think that in
another
the intervention
consideration is.
Go ahead.
Perhaps Mike first.
Sorry.
Mike and then Lisa. Mike.
Yes. Sorry about that. Yeah. Challenges with the virtual format.
Early intervention in type 2 diabetes, there's the opportunity of preserving the beta cells that remain in intact islets prior to destruction. And so when you look histologically at pancreata from individuals at diagnosis, they're often lobules containing islets with, that seem relatively preserved in terms of their architecture prior to the immune system attacking that lobule of the pancreas. Those preserved islets are probably what allow for the physiologic insulin secretion that can be demonstrated with higher amounts of residual C-peptide. And so by protecting those islets early at the onset of diabetes, we are preserving that physiologic islet functionality in terms of not just the insulin secretion, but also the glucagon secretion and the directionality of that, both insulin and glucagon, immediately to the liver to allow for appropriate hepatic glucose regulation of the peripheral glucose levels. And so I think that what we are attempting to do with early intervention is restore islets that are there already, as opposed to having to wait decades and then replace them back.
Yeah, and Lisa, I give the mic to you immediately, but what Mike said is also perfectly aligned with what Carla showed on the sensitivity, the response to hypoglycemia and the correlation she observed. Lisa.
Thank you. Yes, I agree. The islet data is highly compelling and really helpful, and I really enjoyed your talk, Mike. Thank you. From a regulatory perspective, I think in general we expect to see data with more than one intervention method on the specific outcome before we're comfortable using that outcome as a validated surrogate for any given treatment. To explain what I mean just a little further, I might use the example of LDL. LDL lowering could be a consideration as a surrogate for CV risk reduction. But that really came about with a long history of many repeated studies showing that intervention and that outcome showed the expected clinical benefit. And we're currently not accepting LDL for every drug class. It's really for statins. So my point is that while the islet transplantation data is compelling and hypothesis generating, what we would like to see is an intervention that directly impacts beta cells on the outcome to validate C-peptide to predict the clinical benefit with more than one mechanism. And we'd like to see it with a therapeutic beyond islet cell intervention, because I agree with Patricia that there's so many things going on with islet cell that it's hard to know for sure whether C-peptide is really fully capturing the full clinical effect.
Having an intervention trial where you would have C-peptide combined with any other relevant clinical endpoint may be the way to open up what you want to see.
Right. Yeah, we
Yeah.
just need to see it
Peter?
repeated multiple times before we can call it validated.
Yeah. Peter and then Stephen?
Yeah, I think this is exactly the same situation we have in Europe as Lisa just described. For LDLC, we would accept an application for any type of molecule, actually. But what we would want to know is then also how does this drug behave in its terms of cardiovascular safety? That's the target where you want to go. And I think it would be similarly here. If you really want to go into an established surrogate, it should really be shown to be the same or having the same outcome on clinical outcomes if you use different interventions. And obviously that makes the hurdle higher for the first or the second compound coming through. But I think the link in a prospective way followed up would be high for the first applications. And clearly there is a discussion of the unmet need. And I said that's a little bit where we feel that the insulin availability at least makes it possible to treat these patients. And although the data, I was really impressed with the presentation today, the data look compelling in the correlation. It's indeed that intervention that you want to understand, because it's not just the benefit of the treatment, it's also the harms that you need to balance out with that.
Yeah, we're turning in circles, of course, because as long as we don't prove it, we will not have the evidence, and we need something to be able to do the trials. But tomorrow, stay tuned when the people living with type 1 diabetes get also into the picture. It will become very interesting, I guarantee. Stephen.
Yeah, excuse me. I mean, I think as we've discussed a number of times, and I know we've also discussed with you, Lisa, that it's difficult to see how we could conduct a trial where we would see a clinically meaningful difference in whether it's hypoglycemia, time in range, whatever the additional clinical outcome is that's required. As you know, and we'll discuss this, I think, tomorrow, that the numbers required to show a difference, to show a meaningful difference, make doing those trials almost impossible. Because I think, as Carla was saying, we already have patients within the first year or two of diagnosis where they have some residual C-peptide. They have very low rates or lower rates of hypoglycemia. As we move forward with more and more patients on insulin pumps, with CGM and closed loops, we're competing with really good glycemic control. So to actually see that benefit within a relatively short period of time, even within 3 to 4 years, which would be a very long trial with trials involving in excess of 1,000 patients, as we've discussed, we know that we, as a company, we can't do that. It's almost impossible to recruit patients into those trials in those sorts of numbers. And I think our fear is that the field then won't move forward, particularly as improvements in glycemic control and therapies for improving glycemic control really would swamp anything we're likely to see in that time frame. So I think that's the challenge that we really face. I think we appreciate the challenge, but it's difficult to see how we're ever going to overcome that. And I think, therefore, we end up accepting that this isn't an area we would go into. And really what we're looking at is glycemic control.
So I think maybe in the interest of the time, we can move on to some of the questions from the attendees, and maybe the panel can address those. There will be more time tomorrow to talk more about trial design and things like that. And so we can try to take the ones in the order that they... Sure.
You know, one of the...
I just want to comment on a high level, because I don't think it's really important to acknowledge that point. It's very important. The solutions to how to get there, yes, it'd be great to add that to tomorrow's discussion in the interest of time. But I just want to say to Stephen, thank you for acknowledging that. And we do understand, which is why FDA at least is considering the idea of an accelerated approval. Because when you're saying that we're going to need either a lot of patients or a lot of time to look at a clinical outcome, there is a mechanism to still get that product to market before reaching that point. And so that's something that can be considered from at least a US perspective.
So, Patricia, you want me to kick off? I mean, there's a lot of questions that deal with exactly the statement, what is clinically meaningful? And so it's directed to FDA. What would you consider clinically meaningful?
So for clinically meaningful insulin independence, it really needs to be the context of how it's used the patient population and what you would hope to achieve. The person asking the question is referring to, I think, a recent advisory committee meeting, which is beyond the scope of this meeting. This meeting is really focused on how would we deal with new onset type 1 diabetes. So I think it would not be unreasonable for the panel to talk about what maintenance or duration of insulin independence for patients who have a new diagnosis of type 1 diabetes would be meaningful to them.
Carla? You're on mute.
Sorry.
Yeah, you're good. You're good.
Okay, I'm not sure we're talking about the prevention space where it was not having insulin independence. We currently have no therapies that are going to make people's diabetes go away. We're going to, because they have diabetes, right? Because they don't have enough insulin secretion. Our aim is to preserve the insulin secretion until such time we have other methods to replace therapy, their islets. And I want to just say that that's an unrealistic statement to say that we're going to give somebody an immune therapy to preserve remaining function, and all of a sudden they would no longer need any insulin. We're not giving them more beta cells. We are keeping what is there. So I think that's an important comment. The other thing I just want to throw out there, just to be provocative a little bit, imagine if you were evaluating A1C today. Right? We know A1C actually is not super reflective now with the HbA, with the glucose monitoring about what's really happening with glucose control. And we also know that the associations with A1C have some wide variability. So in other words, there are many people who get complications who do not have abnormal A1Cs, et cetera. So I think it's just important to, try to connect the dots here. The dots are controlling glucose is undoubtedly important, but the disease is beta cells. And the pragmatic issues that I think we've heard from Dr. Goff really do indicate we have very strong both correlative data and the intervention data from the islet transplants. Perhaps a commitment to longer term follow-up and see about these correlates, in real life. But otherwise, it seems impractical we will ever be able to crack this nut.
So I mean, I understand. So there's two types of early studies. One is to prevent progression to diagnosis, another, or delayed progression to diagnosis, or intervene early in diagnosis. Well, I guess what I'm asking is sort of, how would you interpret, like, would they only ever need initially basal, and that you would preserve enough endogenous insulin that, the risks of insulin overdose or insulin, to carbohydrate mismatch would be lowered, and that would help protect them from hypoglycemia, or, because they have endogenous insulin, it would protect them from DKA. What kind of clinical measures should, besides just the measure of the C-peptide, and how long would you want to see those, would be informative to how well you thought that C-peptide was correlating or with a clinical benefit?
Could I just take a crack at that? I think Carla emphasized this, and I think I completely agree with it. It's very hard. It's almost impossible to be saying there's a particular insulin dose that is safe, and a bigger insulin dose is not safe, and so on. I think people can develop hypoglycemia at very low doses of insulin. So I don't think it's possible to draw that, to come up with that threshold, frankly.
So what I wasn't suggesting that we would come up with a dose. The concept of having endogenous insulin is that you do not have to give the patient all of the insulin they need for their glycemic management. You would think that the benefit is that you have some level of endogenous insulin that gives you a buffer against insulin delivery error, calculations errors, because you're not getting all of your glycemic control from the exogenous insulin, but the endogenous insulin may be providing that buffer. But how would you evaluate that to figure out this? What we're seeing is the function of the C-peptide, not a function of that patient's strict management of their diet and exercise paradigms, or super users of the 24 basal rates on their pump, or those other things. How would you try to assess this? Because this is where it gets complicated in these studies, is that how are you going to link this to something that is meaningful? And I think this is one of the challenges that we're trying to discuss. And
so I think one of the challenges here is the notion that there is when people get hypoglycemia, it's because they have mismanaged their diabetes. I think that is just incorrect. I think the challenge
I'm not suggesting they mismanage. What I'm suggesting is that devices have errors. Insulin delivery devices have errors. Glucose measurement devices have errors. Carbohydrate counting has errors. So they're not mismanaging. They are doing the very best they can with the tools that we have available to give them. But there are inherent errors, even if they do it correctly.
Yes, and as Dr. Harold pointed out in his presentation, anytime you do not have endogenous secretion, where it's in the pancreatic milieu, where you're getting first pass liver, as Dr. Rickles pointed out, you will never have that same effect. So I think it's important to recognize that, the story of how people are living and coping with their diabetes has gotten super better over the years. But there's a missing component, and that is physiological insulin secretion, which cannot be replaced with exogenous delivery.
Can I just frame it slightly different way?
Kevin.
I think I understand what you're driving at, and it makes sense. It makes complete sense. In other words, if we could know how much of your glycemic control is generated by your endogenous production and how much is being covered for by whatever we're giving with a hybrid pump, for example, that might give us some measure. But the problem is, think about another situation. Think about type 2 diabetes. People need huge amounts of insulin to restore, because of insulin resistance, to restore, some resemblance of normal glucose control, and they still have endogenous beta cell function. So I just think it's extremely complicated and perilous to be able to come up with a measure that reflects exactly what you're asking for. Although I agree with you, if we had some way of determining how much of this is autopilot driven, in other words, driven by your endogenous production versus what you're taking exogenously, that would be ideal. I just don't think we have it.
Can I go back to Stephen Guff? Stephen, I think you proposed a model to include also insulin dose in the whole discussion.
Yeah, so my explanation, and I admit may have been slightly clumsy, and particularly after what Carla was saying about insulin dose. But one of the things that we noticed in our phase 2 study was that we did reduce insulin dose by around 40%. Now, it's then difficult to say, well, is that clinically meaningful? And I think what I was trying to say was that my experience with patients is if you reduce their insulin dose or the lower their insulin dose, the better they often feel for a number of reasons. Now, you're not going to eliminate hypoglycemia. You're not necessarily going to improve their A1C, although I think you would if you had endogenous insulin. But I think the important point is that a reduction in insulin dose is perceived as beneficial by the patient. And what I was also trying to explain was that maybe from an insulin resistance point of view, in the long term, there may be benefits by having a lower insulin dose. So what I was trying to do was to address the issue of what is clinically relevant. Is it important to have a lower insulin dose? I believe it is. But if you said to me, is it a 25% reduction, a 50% reduction? I can't answer that question. But I do think it's important. And I also think that if you are preserving C-peptide and you see a reduction in exogenous insulin requirement, well, then to me, that's showing that what you're doing is working.
Yeah, and again, coming back to the whole principle of type 1 diabetes, it is all about beta cell destruction. If we could measure beta cell functional mass in another way, we would not be sitting here. But that's what we're looking for, any way to measure functional beta cell mass, hence C-peptide. And then I hear also adding any clinically relevant measure. I heard insulin dose and, pick a cutoff. I heard time in range out of the words of Peter Moll with the new measures. Patricia, I know you are allergic to using time in range because of the still underperformance of our tools that we have in the low ranges. Do you wish to comment on that, Patricia? You're on mute.
No, it's a measure that's clinically very useful, but one that also really isn't a validated surrogate for outcomes. So we have to be careful not to use one unvalidated surrogate to support another one. But I think clinically, for management of the individual patient, it's highly helpful in helping them with their care.
I'd like to
get back to Go ahead.
But I also want to point out that talking about CGM, use and everything else is not the topic of this. So we can
no, but we're trying to find other clinically relevant measures to add to C-peptide. Carla,
so I'm just saying, yeah.
Go ahead, finish. I'm sorry.
No, please do. Go ahead.
Yeah, so I guess I'm just harking back to what I tried to put in the study. If you did a very large study like Dr. Goff is describing, because our understanding about the physiology and the intervention data that we heard from transplant, we would expect all these things to be associated. I'm just saying on repeated phase 2 trials, because of the endogenous secretion that people have, the likelihood of repeatedly demonstrating these markers is going to be limited because there's so much of the other variation you don't have enough people in these studies to account for, unless you're going to give everybody the same pump, the same instructions about what they have to eat, etc., which is not realistic way of life. So I'm just suggesting that it's a conundrum here, that the likelihood of robustly doing this in these kind of smaller studies is extraordinarily low. So what we need to be able to say is, we understand the physiology, we understand exogenous secretion is much more, exogenous dosing is going to give you higher peripheral doses, which is likely associated, which we don't have time to get into today, with cardiovascular bad outcomes. So there's a lot of things about preserving secretion. The question is, what's the pathway to go forward so that we can really have this kind of opportunity for patients?
Peter?
And so it also, I respond to that because I think I can immediately straighten or reset a little bit the discussion you had around the CGM. I offered that in my presentation as one of the tools that also needs further validation before we really understand it and the value of it and the link to the clinical outcomes. We think it is something that's worthwhile, but it's also not yet there. So for me, because I'm here also primarily to listen. I mean, you are the experts here at the table, and we want to learn also on where we are at the moment with C-peptide. Obviously, we monitor that we haven't had any regulatory experience with C-peptide and in applications. So we need to understand that the drug has these two sides of the intended and the unintended effects. So what we need is perhaps something like a more in-depth interaction after this meeting, where you could come to the EMA and say, well, this is the material that we have, this is what we present, and that obviously can be done by individual companies coming with their product, but it would be much stronger if you come to this qualification procedure that we have, where we can really have this in-depth interaction with the scientific community and look at the data, also have a more own analysis. I'm not sure if I'm still there, but where we have this possibility of interacting much more in detail on the data that are there, and also to understand what we can ask. I understand that there is this problem of having a very good treatment at the moment available, but that obviously also makes it not easy to say, well, let's lower the hurdle because we have a good treatment available. With all its caveats.
Peter, thank you very much. And again, we acknowledge SOP and everything, but in Europe, I do believe it is a different path. Here, we need indeed to help FDA and our colleagues to reflect on how to use this C-peptide and whether it's up for use. Mark Peakman, I want to come to you, because you're here also representing industry, but your background is immunology. So are immune markers ready also for prime time to add to C-peptide as a package that FDA could look at? One of the first questions of the audience today was, what is disease-modifying therapies? So can we come up with a bigger package?
So to answer, can you hear me okay?
Yeah.
To answer that very quickly, I think it would be great to have a discussion along the lines that Peter suggested, where we could bring that kind of dimension in. And I think something that for a specific agent measured immunological efficacy could be a useful addition to the conversation. However, I don't think it'll solve this. And I feel with the discussion as I followed it that we are trying to push this off into the long grass with thinking about CGM and future possibilities without really addressing, what we have tried and tested over many years. And where I sit, I don't just look at type 1 diabetes as an autoimmune disease. I'm also involved in the development of drugs for lupus, psoriasis, and so on. And there we really struggle because we have very, very subjective measures. We have risk scores. We have combination scores of outcomes that are really hard to nail and be objective around. And my rheumatological colleagues often comment to me how lucky we are in type 1 that we have C-peptide, an objective measure of the target cell that we are trying to protect. And I think the message that I've got from today's discussion is that nothing is really going to beat that. And I would ask for what I'm hearing is just that little bit more of a pragmatic approach here that Stephen and others are outlining, where we don't have to keep waiting for something miraculous to come along, but we say, okay, C-peptide is pretty good. What's the evidence you need? Let's work together to present it properly.
Yeah, and how to standardize everything. I believe FDA
that's a given.
Voila. Excellent. Lisa.
Yes.
Your reaction.
So I don't know if you noticed my video cut out for a minute. Could you just reframe the question, my reaction to specifically?
Well, so I I asked Mark Peakman, now with Sanofi, but responsible for many for many immune diseases, his reaction to the discussion on C-peptide. And so
Yeah.
he really said, this is something you
Yeah.
have in your hands. In rheumatoid,
Right.
we have subjective pain scores, for instance.
So
So should
I
we
do
have our
agree.
foot in the door?
Yeah, I think that we are
Sorry.
lucky that we have C-peptide that represents the pathophysiology of the disease, and that we don't have to rely on subjective measures. The challenge for us is then translating that biomarker change to a clinical benefit. And I think that I think I'd like to emphasize the reason we need to do that from a regulatory perspective is because when we make yes or no decisions about a drug, we need to weigh the benefits and risks. And all drugs carry risks. And so if we don't really have a good sense of the quantifiable benefit of the drug, it's really difficult to weigh it against the risk. So just to emphasize, our role is to make yes or no decisions, in addition to helping facilitate drug development. But at the end of the day, when we have an application in front of us, it's not just enough to know that C-peptide was increased versus some comparator or placebo. We need to know what that increase means to patients because we have to balance that against the risks of the drug.
Okay. I believe that we have, in a very erratic way, taken all of the suggestions of the audience on our different aspects. So, and I apologize because there is a delay apparently over the ocean when I talk to you guys. So I want to give every one of you final last words. And then I will hand over to Patricia to make her final comments. So I will take you in the sequence that you have talked. First of all, Kevin, final words.
So I completely understand Dr. Yanov's point. I think that's it's somewhat it's reassuring that this is the approach that's being taken to evaluate drugs. I think it makes complete sense, and like I said, it's reassuring for patients. On the other hand, I think that the data that was presented this morning provides a compelling story about basically the idea that C-peptide is measuring the disease, and that we have a number of very good assessments of what its significance is. First of all, we have the data that Mike presented that restoring C-peptide provides clinical benefits. If nothing else, I think the data across all the different trials, including the associative data from the DCCT, from the Scottish data, and even from some of the intervention trials as well, would suggest that preserving C-peptide reduces hypoglycemia as one measure of clinical outcomes, and it may actually have additional benefits as well. It's challenging to measure that, as Dr. Goff pointed out, because if we're talking about people in the first few years of the disease, they still have significant residual beta cell function. So it's hard to tease apart where the decline in beta cell function, what effects the decline in beta cell function would have at those very early stages, which is the time where we have to intervene, because that's when patients still have the residual C-peptide that we think is going to be clinically important in the long term. So it seems to me that we've, kind of fulfilled what the criteria were laid out in terms of the consideration of accelerated approvals with the data that's already available.
Mike?
I think Kevin gave a brilliant summary of the discussion, and I would echo that, and that I believe there are sufficient data in early intervention as well that preserving C-peptide has had important effects on glycemic control, lowering insulin requirements, and protecting people from severe hypoglycemia, but that to require that in every trial of a new intervention would be prohibitive, but there's clear relationship of that preserved C-peptide on these clinically important outcomes.
Carla?
First of all, I want to say to my regulatory colleagues, I'm glad you have your job and I have my job. You have a tough job. So I do appreciate that conundrum you're in, and I appreciate the emphasis on weighing the safety with the uncertainty of what level and what duration of C-peptide and for what benefits. I understand the conundrum you're in, and I'm just thinking about this conundrum from the other side, which is, how else are we going to do this? We have such promise here to change the course of the disease. And that's really, we have great evidence, reproducible evidence. And I think that when we get these therapies in practice and more people can avail themselves of them and we commit to long-term follow-up, we could continue to have the opportunity to look for these significant benefits, and that we don't think about this disease as a metabolic disease. We think about this disease as a destruction of the beta cell, which is having all these downstream consequences. So anyway, I hope that this is helpful. I'm always welcome to discuss data as much and as frequently as possible with regulators as needed. So thank you.
Stephen?
Yeah, so first of all, thank you for allowing me to be part of this discussion. And I will also start off by saying, I fully appreciate the FDA's position and what Lisa is saying, and I really do. I get the bit about benefit risk, and as a doctor, of course, I understand benefit risk. I think my fear is that if we have to rely or if we have to demonstrate glycemic parameters in this area, I think it will prove a problem, and unfortunately, more companies will walk away from this, and I think we'll struggle to make progress in what I think is an important area of trying to preserve endogenous insulin secretion. And I will say from a company perspective that actually, from a commercial point of view, this isn't a big deal. This isn't something that earns companies like Novo Nordisk or likely to earn money. I mean, this is something that we have committed to as a societal responsibility more than a commercial deliverance. And my commercial colleagues probably give me as big a battle internally within Novo Nordisk as the FDA give me externally. So it's always a difficult one for me to fight. I think though that I take comfort from the accelerated approval with C-peptide being seen as a reasonably likely surrogate endpoint. I think what I might ask for is a bit more flexibility beyond that. Can we look at ways beyond a traditional phase 3 study with thousands of patients where we can ultimately deliver some clinical benefit, whether that be through some form of pretty strict registry or whatever, so that we get an accelerated approval, we get it used, and then we provide you with some clinical data. Because I think if it's going to rely on a phase 3 trial, I know from our point of view, it's almost certainly not going to happen.
Yeah.
May I respond?
And finally, yes, Lisa, please.
Oh, I'm sorry. I thought.
No, Lisa, go ahead.
Okay. Well, I just want to again say how robust and excellent the talks were. I personally gained a lot from listening to it. I would concur with Dr. Harold. I think we are at the point where accelerated approval standards have been met, and we are open to that with the data that have been presented today, as well as things that we've been considering in the past. We want to make sure that treatments that reach patients have been scientifically, robustly evaluated, but we certainly don't want to stand in the way of progress and having those therapies reach patients as soon as possible. And I think that's why the accelerated approval pathway, it may really serve both functions very well. While I know that the recent Alzheimer's drug approval is very controversial, and I'm not going to get into the merits of the action there, I will note that the confirmatory trial that I believe is required, I read is the company's been given 9 years to complete it. And so I think what we need to understand is that we're not asking for these trials to be done necessarily within 1 or 2 years with thousands of patients. It could be that long-term follow-up of the smaller little cohort is sufficient. I'm also intrigued by some of I honestly am not sure if it was in today's talks or some of the talks from tomorrow that I've listened to ahead of time, but the idea of how well understood the natural history of C-peptide decline is, and whether we might be able to use that as a comparator to limit the enrollment of patients into placebo arms. So still generating that robust data, but without having patients who really are at risk for this terrible disease not have to wait a long time to be able to have access to these therapies, even if under IND or under an experimental protocol. So again, FDA does appreciate the challenges and does want to work with companies and stakeholders to facilitate drug development in the space as much as possible.
Mark, did you want to add anything?
No, as the immunologist outlier, it would be completely inappropriate for me to call judgment on this with the final comment, except to say, I don't think I've heard this case better presented, and I've been at many of these discussions over the years than today. I thought Kevin's summary was on the money, and I thought Stephen's summary of the industry position was equally on the money, and it's very nice to hear the very positive comments coming from the FDA.
Peter, we as Europeans, happy to be part of this?
Well, surely learn a lot of the same concerns, obviously, as the FDA has. We have a little bit of a different public health system, so we're also always listening to our colleagues in the payers in the health technology assessment. But at the same time, to the clinicians, in the preparation of this work, we talk to them, and they really ask what the patient feels, and experience is obviously very, very dear to them. C-peptide, obviously, as you presented eloquently today, I think has a very strong rationale, and the correlations look also good as far as I saw that information that you presented clearly. But the path forward, and then Stephen suggested a bit of what that could look like. Lisa gave also some suggestions what all these could be. They're all less than the standard that we normally apply to drug application. And so it will be something that we really have to look back on, and on the specific drug also. I think also really the patient's voice tomorrow. I will be very interested in hearing what they express, and what could they see in addition, what do they feel of the C-peptide. And I think that that's going to help and shape my discussion also with my colleagues.
Excellent. Patricia, all yours.
Okay, so I'd really like to thank the panel and the presenters for really well done presentations of decades worth of knowledge, and for very robust discussions. It is going to be a challenge to look at all this varied information and look for common factors. And as Lisa says, our goal is to understand it. So when we make a determination for products that can improve, patients' health and well-being, that we can give confidence that the potential benefits of the therapy and what we've demonstrated are well balanced with whatever the risks are of that treatment. And we want to be careful in that, but we do not want to be so careful that nothing moves forward. So you've given us a lot to think about. Tomorrow, I'm sure the discussions will give us even more to think about. And thank everybody for their time. And we hope that we addressed the majority of the questions to the attendees. And we couldn't get to everything, but have a good rest of your day. Take care.
And
Hi there. I think that cut out there just as Chantal was about to say goodbye to everyone. But just wanted to circle back at the closing this out, and just from the c-path side of things, of just being able to watch from the sidelines as the conversation was going, and thank all the speakers for day one and the passionate and compelling work that was presented. I think like any good story, it started off with a description of the setup, which was the unmet need, and we got into the conflict side of things, which was, on some parts just a description of terms, realizing that people who've dedicated their life to work to modify this disease progression can use terms that are different than our colleagues in the health authorities. We see that every single time at c-path, every single time in every therapeutic area, and that's okay as long as we can focus on understanding that we are using terms in ways which might carry different meaning. And ultimately, where to the last act we started getting there is the idea of what is resolution. And we're going to be reconvening tomorrow, where we start to dive into a little bit more on the clinically meaningful measure side. But it's interesting to hear colleagues, from the FDA talking about mechanisms such as the accelerated approval program and the reasonably likely surrogate endpoint, and our friends from the EMA talking about ways of resolution which might be further dialogue around specific points. As I said at the opening of today, we weren't going to solve all the needs in T1D drug development in 2 days, and we certainly didn't do it in one. But I think being able to understand how and why we use terms, and such as validated in certain contexts and what meaning they carry, is something that when we become mindful together, we can actually get down to the nub of the issues and think about what solutions we can move forward with in a data-driven fashion. So that's it for today's side of the workshop, and we'll be reconvening tomorrow, where we'll be taking up session 2 and 3. So thank you again for making the time to join, and really again, thanks to everyone for putting into the questions into the Q&A, where they were coming thick and fast. So very much appreciative for your time today, and look forward to reconvening tomorrow. Thank you so much.